All right, today we will be covering chapter 2, which I'm sure will be everyone's favorite chapter. It deals with chemistry. All right, so as always, some roots, prefixes, and suffixes that we will be talking about throughout the chapter. Bio just means kind of life. Di, two, mono, one, poly, many. Sacra means sugar. Lipo, like a liposuction. Fats. Iso means equal. All right, so um, why do we study chemistry? Well, because we have to. Um, Pretty much everything in anatomy and physiology is related to chemistry. Um, the true definition, chemistry is concerned with the composition of substances, properties, how they change in chemical reactions, and the interactions of matter. So when we look at, the human body, food, medications, they're all composed of chemicals. We talked last chapter about, atoms, compounds, molecules, stuff like that. And when we look at chemicals and chemistry, we see this in how digestion occurs. We break down our proteins into amino acids. We break down carbohydrates into sugars. We break down lipids into triglycerides and fatty acids. And then we use those nutrients to provide cellular energy. Remember, I said that oxygen, we don't need oxygen to sustain life, but we use oxygen to release that energy from the food that we eat. How medications are absorbed, all is chemistry based. Obviously, all of our anatomical structures are chemicals. We can actually draw the chemical structure of actin, which is one of the components of skeletal muscles that we use for contraction of skeletal muscles. All physiological processes are based on chemical reactions. When we look at in chapter 4, we'll be talking about glycolysis and how we break down glucose into pyruvate. It's based on chemical reactions. Water, H2O, electrolytes, they make up the body fluids. Minerals, proteins, they are going to make up our bones. All right, so matter. Matter is anything that takes up space and has mass. Matter is composed of elements. We talked about the elements. That's what you find on the periodic table: hydrogen, oxygen, nitrogen, carbon, which in turn are composed of atoms. Atoms have electrons, neutrons, protons, and they can be either solids, liquids, or gases. Water is a liquid, ice is a solid. When you boil water, it turns into steam. That's a gas. We're not going to kind of differentiate between mass and weight, even though they are two different things. Mass is actually measured in pounds. So typically we say, oh, I weigh 170 pounds. But technically that's not correct. Technically we should say my mass is 175 or 170 pounds. Whereas weight is due to the gravitational pull, and it's actually measured in this thing called newtons, which if you take chemistry, you'll learn about newtons there. So your mass is kind of relative to your weight based on the gravitational pull. So if you're on a different planet, you would have a different mass. For example, my mass on Pluto is 10.8 pounds. My mass on Jupiter would be 421 pounds, and my mass on the moon, Earth's moon, would be about 30 almost 31 pounds. All right. Chemistry, study of composition, properties, and interaction of matter. That's the focus of this chapter. Biochemistry is basically the study of biological chemistry. For those of you bio majors or chemistry majors, you might have to take biochem. Basically, it's going to be the studies of physiological processes in disease, and it kind of mirrors pathophysiology if you nursing majors will be taking that. All right, so here's the periodic table. The good news is that you're not really going to have to memorize the periodic table. Pretty much what we see in the human body are going to be hydrogen, lithium, sodium, potassium, magnesium, calcium, maybe some oxygen, carbon, nitrogen. We'll see those, maybe a little bit of chlorine, iodine. So really those are going to be the elements that we really deal with in biology or in this class. So we'll be coming back to reference this, this periodic table throughout this lecture. All right, so elements, simplest type of matter. We have 118 elements on. We have 118 elements on the periodic table. You know, iron. Iron is iron's an element. Sodium, potassium, oxygen, hydrogen. 92 of these elements are naturally occurring. The remaining elements are not naturally occurring. And we combine these, if you remember from last chapter, we combine these elements into compounds. For example, sodium chloride. We take sodium, Na, and chloride, Cl, and we put them together and we get sodium chloride, which is salt. We take glucose, right here, 6 carbons, 12 hydrogens, 6 oxygens, throw them together, make glucose, sulfuric acid. And then I just threw this one in here. This is actually called Titan. You don't have to memorize it, but it's actually made up of about 34,350 amino acids. It's actually 244 approximately folded proteins together. If you actually tried to pronounce its name, it would take you over 3 hours to pronounce. So we just shortened it to Titan. Atoms, those are going to be the smallest particles that we talked about last chapter. You know, protons, neutrons, electrons. Different elements are required by the body in different amounts. We have bulk elements. They're required by the body in large amounts. We need lots of carbon, lots of oxygen, lots of hydrogen, lots of nitrogen, sulfur, and phosphorus. Remember, phosphorus is in our bones. Trace elements required by the body in small amounts. Iron and iodine. Ultra trace elements are found in very minute amounts, such as arsenic, which I think is funny because arsenic is actually can be used as a poison. So here's a little chart just kind of breaking down the major elements, the bulk elements, the trace elements, and the ultra trace elements. So you can see here that here are the things that we find in our body that make up, you know, less than 0.1%. Here are the things that make up, you know, about 99.9% of the body. But if we look at, you know, the top 4 elements, carbon, hydrogen, oxygen, and nitrogen, they're going to make up 96.2% of what we find in our body. So greater than 95%. So carbon, hydrogen, oxygen, and nitrogen. And I just remember CHON, Chon. Don't know why. But CHON, the 4 major the four major elements that we find in our body. And as we go through the different types of components of our body, we'll see these. For example, proteins have amino acids make up the building blocks of protein. So proteins have carbon, hydrogen, oxygen, nitrogen. Fats have carbon, hydrogen, oxygen. Fats are lipids. Let's see. DNA, RNA, carbon, hydrogen, oxygen, nitrogen. All right, moving on. Okay, so when we look at the what we find in an atom, we see that the atom is the smallest property of an element. So we have an atom of carbon, an atom of hydrogen. And we know that the atoms contain protons, electrons, and neutrons. So an electron is something that contains a negative charge. And we'll see examples of these later on. Electrons contain a negative charge, and they really don't carry any weight to them at all. When we look at the structure of an atom, we'll see that we have a nucleus of an atom, and then the electrons don't reside in the nucleus. The electrons are kind of floating in orbit around the nucleus. Then we have protons, which carry a positive charge, and they are found within the nucleus. Neutrons carry a neutral charge. Neutrons and protons roughly weigh the same, and neutrons are found within the nucleus as well. All right, so atom, protons, neutrons, electrons, protons, and neutrons are within the nucleus. Electrons are orbiting around the nucleus. Protons and neutrons roughly weigh the same. Electrons pretty much have no weight to them whatsoever. Now, we also have these things called ions, which we will get to later on. Ions are basically when an atom either gains or loses an electron. So if you ever see something like Na plus or Cl minus, that atom has either gained or lost an electron. Like I said, we'll see examples of that later. And a molecule is just, as you already know, when we combine two or more atoms together. All right, so atoms composed of subatomic particles, protons, neutrons, and electrons. If we go back, okay, so the number of protons determines what the element is. So it says, example, sodium has 11 protons, potassium has 19 protons. So if we go back to our periodic table, sodium has 11 protons, potassium has 19 protons. If we were looking at chromium, it would have 24 protons, cobalt has 27 protons, copper has 29 protons. So that's how we get the number of protons. The number of protons determines what the element is. Always, always, always. There's never an exception to that. Now, remember that the nucleus contains both protons and neutrons. Remember that neutrons carry no electrical charge. Remember that protons and neutrons roughly weigh about the same. Remember, protons have that positive charge. and that electrons carry that single charge. Electrons, remember, electrons orbit around the nucleus. They are not part of the nucleus. And if you've ever taken chemistry class, in chemistry, you learn about the sp, d, and f orbitals. The 1s2, 2s2, 2p6. We're not going into that in this class, but electrons are responsible for energy levels, which is that 1s2, 2s2, 2p6. Like I said, we're not going into that. So if the number of protons equals the number of electrons. Remember, protons are positively charged and electrons are negatively charged. If the number of protons equals the number of electrons, we would say that that atom is electrically neutral because the positives equals the negatives. We don't have, overall, we have a zero charge. If the number of electrons outweigh the number of protons, then we would say that it has a negative charge because the electrons have a negative charge and they outweigh the number of protons. If the number of protons outweighs the number of electrons, we would say that that element has a positive charge. And remember, those are called ions. Ions are formed when an atom has either gained or lost one or more electrons. But if the number of protons equals the number of electrons, we would say that that atom is electrically neutral. All right, so let's look at lithium. So lithium, we go back to our periodic table. Lithium has 3 protons. So we can count here. Remember, protons have a positive charge. 1, 2, 3. 3 protons. Okay, so we have 3 protons here. And then we have this little 7 that's up here. So we would actually count 1, 2, 3, 4 neutrons, because remember, neutrons lie in the nucleus as well. So we would actually have 4 neutrons. So we have 3 plus 4 would equal 7. So that's how we would get that 7 that's up on top. 3 protons plus 4 neutrons equals 7. That's how we get that 7 up there. And then lithium, going back to our periodic table, that symbol for lithium is Li. So that's how we get this chemical symbol right here, Li. So we know that, or we can see that we have 1, 2, 3 electrons. So because we have 3 protons and 3 electrons, we can say that this atom is electrically neutral. Remember, the number of protons equals the number of neutrons, so this atom is electrically neutral. All right. Now, kind of why are these numbers important? So we have these things called atomic number and mass number. So the atomic number just represents the number of protons. So let's look at nitrogen. If we go back to our periodic table, nitrogen is the symbol N, and it has 7 protons. So we have our chemical symbol N, and we have 7 protons. So remember, each element has a unique atomic number because the number of protons determines what element it is. Now, And then we've already been through this example before. Sodium has 11 protons, potassium has 19 protons. The mass number is the number of protons plus the number of neutrons. And we saw that here in the lithium example. The number of protons, 3, plus the number of neutrons, 4, equals this number up here. So we can work backwards and say, how many neutrons do we have? So we can say 14 minus 7. So we would say that this element, nitrogen, has 7 neutrons. 7 plus 7 equals 14. So remember that neutrons don't carry any charge. Protons carry a positive charge. Electrons carry a negative charge. Also, we know that this is an electrically neutral element because there is no plus right here, or there is no minus right here. Since there's no plus or minus, we can say that the number of protons equals the number of electrons. So because we have 7 protons, we could also say that there are 7 electrons in this nitrogen molecule. Now, remember that I said that protons and neutrons have a weight, but electrons really don't weigh that much. Electrons really don't contribute to the mass because they are so light. Electrons only weigh about 1/2000th of the mass of a proton or a neutron. And if you really want to know, a proton weighs about 1.67 times 10 to the negative 27th kilograms, which is about the same as the mass of a neutron. Again, it's still very, very, very small. It really doesn't weigh that much, but so there's your fun fact for the day. Now, atoms of different elements differ from one another because they have different atomic numbers and different mass numbers. So remember that the number of protons make the element different. So they're going to have different atomic numbers, and then they will have different mass numbers as well. All right, so let's work through an example. If gold, which is Au, were to gain a proton, what would it become? So it gains a proton. Remember, a proton is a positively charged ion. So we would have, if it gains a proton, what would it become? So we could throw out A because this little symbol right here means it gained an electron. So this answer B, we would never write something like that. So we would throw that one out. This one is a viable answer because it looks like it gained a proton. But what about D? Mercury. So we're between C and D. So we're going to throw out C. D is going to be the right answer because remember, the number of protons determines the element. So if something gains a proton, it actually becomes a different element. So we would actually, if something gains a proton, it actually becomes mercury. 79 plus a proton equals 80, which is mercury. So D would be the correct answer. Now C, what happens with C is it actually lost an electron. That's why there's that positive charge right there. Now I can tell you gold cannot gain a proton to become mercury. That's, if you've heard of alchemy, that's what alchemists tried to do many years ago, was to try and convert something into gold. They tried to convert, silver into gold, lead into gold, some other metal into gold. They tried to change the number of protons into gold, which we really can't do. So this is just kind of like a hypothetical question. All right, so we're going to do another practice problem. All right, so let's say we have an atom with 82 protons and 125 neutrons. So it's a neutral element. How would you write the atomic number and mass number? So we know it's a neutral element. All right, so we go to the periodic, we know that the number of protons dictates what that element is. So we go to the periodic table and we see what element is listed as 82. So that's going to be lead. So 82 is the number of protons. So that's going to be the atomic number. That number is going to be on top. Or that number is going to be on the bottom. So let's write our PB 82 right there. And then it says, so that number goes on bottom. Then it says the mass number goes on top. So how do you calculate the mass number? It says that we have 125 neutrons. But the mass number, which goes on top, is the number of protons plus the number of neutrons. So 82 plus 125 is 207. So there. And then it says this element is a neutral element. So we have 82 protons. So we would know that we have 82 electrons. So we don't have any positive or negative charge out here. So basically we are done. 82, 207, Pb. Pb is lead. So that's what we would write, and that's our answer. All right, here's another one. What is the mass number of an element whose atom contains 8 protons, 8 electrons, and 8 neutrons? So mass number. Remember, atomic number versus mass number. So we have mass number is the number of protons plus the number of neutrons, which is 16. So B is going to be our answer. All right, so we also have these things called isotopes. Isotopes are atoms that have the same atomic number, but different mass numbers. Basically, they just have different numbers of neutrons, but they have the same number of protons. And we see this with oxygen. Oxygen can have, remember, if we go back to our periodic table, actually, if we just look at here, we know that oxygen has 8 protons. So here, oxygen would have 8 protons and 8 neutrons, 8 protons and 9 neutrons, 8 protons and 10 neutrons, 8 protons and 20 neutrons. Radioactive isotopes, uranium, for example, they're very unstable and they're going to decompose and they will release energy. If you look at like the atomic bomb, they release atomic radiation until they gain stability. And we actually use some sort of radioactive isotopes to detect and treat disease. Some thyroid diseases, for example, are used. We use radioactive isotopes to detect and to treat thyroid disease or thyroid cancer. Now, it says here that the atomic weight is considered the average of the mass number of all isotopes. So what does that mean? So basically, if we go back to our periodic table, we look at, let's look at zinc, for example. We look at this weight right here. This weight would be the average number of all of the isotopes combined. Thallium, this number right here is the average weight of all of the isotopes combined. So really, all of the elements in the periodic table, they don't have really an even number of weight. It's not like any element has a number 0.000000. So we can see that pretty much every element has isotopes. You look at these elements down here that have all of their weights kind of in parentheses. These are non-naturally occurring elements, so we really don't know much about them. So we really can't calculate their weight. So we're just kind of guessing at their weight was, or we don't know what their stable isotopes are. We can't determine their weight because we can't look at their isotopes long enough to determine what their weight is. All right, so then we have the statement, because the way atoms interact is due to the number of electrons, isotopes of an atom chemically react in the same manner. So if you take organic chemistry, you will learn that the electrons are what makes or breaks the chemical reaction. Electrons do the moving in a chemical reaction. So therefore, isotopes of an atom are going to chemically react in the same manner. The electrons are going to be what is running that chemical reaction. All right, isotopes. Which of the following pairs of atoms are isotopes? 6 protons and 6 neutrons versus 7 protons, 7 neutrons. 6, 6 versus 6 and 7, 6 protons, 6 neutrons versus 7 protons, 7 neutrons. So go back to the definition of what an isotope is. An isotope is the same element, but different number of neutrons. So this one is a viable answer because they have different number of neutrons. This one's a viable answer because this has a different number of neutrons. This one does not have a different number of neutrons, so we can cross that off. But isotopes also, they're the same element. So we need to pick the one that has the same number of protons. 6 and 7. That one's out. 6 and 6. This is going to be the same element, but different number of neutrons. So this one is going to be an isotope. So in answer A, this one is answer A. This one is carbon. This one is Oh, that's a bad N. That one's nitrogen. And answer B, those are both isotopes of carbon. And then in answer C, that one's carbon and that one's nitrogen. All right, so I said that radioactive isotopes can be used to treat diseases, and I mentioned the thyroid gland, um, only because it metabolizes iodine very well. Iodine's a good not necessarily non-harmful substance. It's not like uranium. Many radioactive isotopes are used in medicine for, coronary blood vessel disease, kidney function, hormone concentration, bone density. All right, when we get to ionizing radiation, if we're um, talking about like nuclear war or something like that. We have different types of radiation. Alpha, beta, gamma, those are all called ionizing radiation. Basically, this radiation can remove electrons from atoms and form ions. Remember, ions is when atoms either gain an electron or lose an electron. And those free electrons would eventually destroy nearby atoms. Gamma radiation is going to be the worst type of radiation. It can penetrate matter very deeply. We use, if you ever take physics lab, you'll be dealing with alpha radiation. It's relatively non-harmful. Alpha radiation is used in a lot of different things, such as smoke detectors, or if for treating metastatic bone cancer. Beta radiation is 100 more times powerful than alpha radiation. It's used in eye and bone cancer. Gamma radiation, we actually use that in the operating room for sterilization of medical equipment, or as food passes through a conveyor belt, we can use gamma radiation to destroy any bacteria on food products. And we also use gamma radiation for cancer treatment, but it can break up DNA. So, you don't want to be exposed to gamma radiation all over your body. All right. So when we talk about molecules and compounds, in this class, we're going to say that they are the same, even though they are not. That's a chemistry class. Molecules basically are when we take atoms and combine them. They're going to be electrically neutral. They're going to share. They're going to share electrons. Methane, CH4, water, H2O. You can see the other examples there. Molecular formulas. We are going to write these in their chemical equations. H2 is 2 molecules or 2 atoms of hydrogen. Now we say it's a molecule of hydrogen. When you have the same element, we say that it's a diatomic element. When we have that glucose, the C6H12O6, now we say we have a molecule of glucose. We have a molecule of water. We have now 2 hydrogen atoms plus an oxygen atom. We have a molecule of water. All right, chemical bonding. Basically, chemical bonds happen when atoms combine with other atoms. It's because of the interaction of those electrons. Remember that I said that electrons drive chemical reactions. Remember, electrons are not in that nucleus. They're orbiting the outside of the nucleus. Now, you can see the picture of argon here, and you can see that there's kind of 3 rings outside of the nucleus. We call those shells. Now, this first shell, this inner shell right here, this first shell can only hold 2 electrons. Always, always, always. This second shell can only hold 8 electrons. Always, always, always. This third shell can only hold 8 electrons. Always, always, always. And then as we get into like the fourth shell, and we're not even getting into the fourth shell in this class, then we can get up into it can hold 18 electrons. And this is when we get into that the VSEPR, valence shell electron pair repulsion. Valence shell pair electron repulsion. The 1s2, 2s2, 2p6, and all that stuff. And basically, the electrons in the outermost shell of that atom are going to react with other atoms to form chemical bonds. So basically, we're going to look at how many atoms, how many electrons are in that outer shell, and that's going to determine whether or not it is going to react with another atom. All right, so if we look at hydrogen, hydrogen is listed in that very top left-hand corner of the periodic table, so it's listed as number 1. So in an electrically neutral hydrogen atom, it's going to have 1 proton and 1 electron. So it's going to have 1 electron in its outer shell. Helium is listed as number 2 in the periodic table, so it has 2 protons and 2 electrons. Lithium, we already looked at, it has 3 protons in its nucleus and 3 electrons orbiting. Remember that it can only have 2 electrons in its inner shell, and then it will have 1 electron in its next shell. Sodium has 11 protons and 11 electrons. Remember, 2 can only be in its inner shell, and then we have 8 in the next shell. So we have 2 in the inner shell, 8 in its next shell, and it says 11 electrons. So then we're only going to have 1 electron in its outer shell. All right, so what is the maximum number of hydrogen atoms a free carbon may bond to or bond with? So carbon's number is 6, so it has 6 protons, which means it also has 6 electrons. So if we have carbon, which is 6, we would have 2 in its first shell, and then 6 minus 2 would be 4 in its next shell. So 2 plus 4 is 6. So our answer would be C. Now, what about hydrogen? Hydrogen only has 1 proton, 1 electron, so it only have 1 electron in its outer shell. So hydrogen could only bond with 1 other element. Well, oxygen. Oxygen is 8, so oxygen 8. So it would have 2 in its first shell and 6 in its outer shell. Actually, I made a mistake. Oops, up here. It can only bond with 4. Carbon can only bond with 4. Oops, my bad. I'm sorry. because we have 4 up there. All right, so oxygen, 2 plus 6 equals 8. So 6 would be the answer for oxygen. So that's how we determine the number of free hydrogen atoms oxygen can bond to, or carbon or hydrogen. All right, boron. Its innermost shell is full. So boron is number 5 in the periodic table. So its innermost shell is full. It has 2 electrons in its inner shell. It has 3 electrons in its outer shell. What would happen when this atom forms a chemical bond? A, boron will lose its electrons from both its innermost and second shell. So answer A is it will lose 5 electrons. Boron, answer B, would gain 5 electrons. So 1, 2, 3. Boron would gain 5 electrons in its outer shell to make 8. 8 is stable, so 8 electrons in its outer shell would make it very stable. Boron would lose its 2 electrons from its inner shell, or boron would lose its 3 electrons from its outer shell. So we have to think about what is going to be the most easiest to do, or what would require the least amount of energy. So let's just look between A and D. Losing 5 electrons versus losing 3 electrons. Losing 3 electrons would require less energy than 5. So we're going to get rid of A. Now, if you remember, we talked about the outer shell only. Only look at the outer shell. So we can throw out C. Boron will lose all its electrons from its innermost shell. We don't touch the innermost shell. We only look at the outer shell. Now again, go back to what is more feasible or what requires less energy. Gain 5 electrons or lose 3 electrons. Gain 5 electrons is going to require much more energy than losing 3 electrons. So D is going to be our answer. All right, so everything that we've been talking about losing electrons, gaining electrons, those are called ions. Remember, an ion, it's an electrically charged atom that has either gained or lost an electron. And we can classify, we can break an ion down into either a cation or an anion. A cation is a positively charged ion. And that happens when that atom has lost an electron or multiple electrons. Sodium becomes Na+. Calcium has become Ca2+. A negatively charged ion is called an anion. And that happens when atoms have gained an electron or gained multiple electrons. An example would be your chloride, Cl-. Now, if we go back to the periodic table, if we look at kind of here, everything right here will, in that first column, will or could form a +1 charge. So H plus 1, Li plus 1, Na plus 1, K plus 1. Everything in this second column. Ca plus 2, Mg plus 2. And we're going to skip this whole middle section. We're actually going to skip this whole section. Everything, and we're not going to talk about this column 18 right here. Those are our noble gases. Those already have those outer shells filled with 8 electrons. So here, everything here, those are going to be your minus 1s, Cl minus, F minus, Br minus, I minus. So really in biology, those are going to be the only ones that we talk about. All right. So because we have ions now, Na plus, Cl minus, when those come together, those are called an ionic bond. We have different types of bonds. We have covalent bonds. We have polar covalent bonds, nonpolar covalent bonds. We have ionic bonds. When an ion and an ion react, those are going to form an ionic bond. Oppositely charged atoms are going to bond together. They're attracted to each other. In the example, sodium plus chloride equals sodium chloride. That sodium ion plus that sodium cation plus that chloride anion is going to form sodium chloride. All right, so what's the difference between calcium, elemental calcium, and Ca2 plus? So remember, an ion is either it is, we can immediately throw out 2 of the answers. Because remember, the definition of an ion is an element has either lost or gained an electron. So we can immediately throw out A and B. Because if it has lost a proton, that becomes a different element. So we're left with C and D. So Ca2 plus means that it has gained a positive charge. Positive charge means that it has lost 2 electrons. So answer C would be our correct answer. Now, if we went from oxygen to O to minus, that's when we could say that gain 2 electrons would be our correct answer. That'd be an example of gaining 2 electrons. All right, so remember that I said electrons form the chemical reaction. So this is what I'm showing when electrons form that chemical reaction. The electrons do the moving. We have sodium and chloride forming that ionic bond. That attraction to each other. Here we have elemental sodium where we don't have any charge. Chloride doesn't have any charge. Now that that chloride has taken on that Cl minus. We have that extra electron that we didn't have before. Sodium has now lost that electron, so now we have Na plus and Cl minus. And kind of what we see now is that the sodiums and chlorides are going to bond together in an ionic bond. The sodiums and chlorides are going to line up together. Remember, like you played with the magnets in second grade and you tried to push the magnets around and the magnets moved because of the positive charge. And if you lined up the magnet positive to positive, the electron or the magnets moved. But if you lined up the positive end to the negative end, the magnets kind of got stuck together. That's what happens with sodium and chloride. You can see that the sodiums and the chlorides are going to be right next to each other because of that positive and negative interaction. You don't see all of the green balls lumped to one side and all of the purplish colored balls lumped over on the other side. You're going to see a mixture of positive, negative, positive, negative, positive, negative, positive, negative, because they're going to be attracted to each other. The positives are going to be right next to the negatives. The positives are going to be right next to the negatives, and so on. Remember, that's an ionic bond because we're taking a positively charged a positively charged ion, a cation, with a negatively charged anion to make a chemical reaction. All right, so what happens when you take the ionic compound, sodium chloride, and place it in water? A new covalent bond will form between sodium chloride. Sodium chloride will dissociate into sodium ions and chloride ions. And the bond between sodium chloride will become a hydrogen bond. Sodium and chloride will each form a covalent bond with water molecule. All right, so one, we haven't even talked about covalent bonds yet. We haven't even talked about hydrogen bonding yet. So based on that, you can actually throw out A, C, and D. B is going to be the answer, but you should already know the answer. Maybe not in chemical terms, but what happens when you place table salt in water? It dissolves. So sodium and chloride is going to dissociate. It dissolves. That's what dissociate means. It's going to dissociate. It's going to dissolve. So we'll talk about covalent bonds here in the next slide. But covalent bonding is very different from ionic bonds. So we're gonna throw out anything with covalent bonds. Hydrogen bonding is another type of bond, which we'll talk about later. So it's going to dissociate. Dissociate just kind of means dissolve. And that's actually what we saw up here. It's going to dissociate or dissolve into these sodium and chloride ions. All right, so covalent bonding. Covalent bonds are formed when atoms share electrons. So if we remember in ionic bonds, we have a positive charge and a negative charge. With covalent bonds, we don't have that positive charge and that negative charge. With covalent bonds, they share electrons. So we're going to start out with neutral atoms, and we're going to end up with neutral atoms. So this hydrogen atom, that's a neutral atom, that's a neutral atom, and that's a neutral molecule. We have hydrogen, remember, is number 1 in the periodic table. It's in that far upper left-hand corner. So it has 1 proton, 1 electron. This hydrogen, both hydrogens have 1 proton, 1 electron. But when we combine them into H2, now we have 2 protons and 2 electrons. But the electrons are shared between both molecules. So basically, if we cover up this, erase. If we cover up this molecule of hydrogen, this hydrogen molecule right now, the hydrogen molecule on the left has 2 electrons. And if we cover up this hydrogen molecule, then this hydrogen molecule on the right has 2 electrons in its outer shell. Remember, 2 electrons in that first shell makes it stable. And that's what we want. So this hydrogen molecule, or this hydrogen atom on the right-hand side, is electrically stable because it has 2 electrons. All right, so let's look at water, H2O. So we've already looked at the stability of these 2 hydrogen molecules. And if we look at oxygen, remember we did oxygen. Oxygen has 8, so it has 2 in its outer shell, or 2 in its inner shell and 6 in its outer shell. When oxygen combines, if we look at O2, 2 molecules of oxygen together, we're going to see that that there's still 1, 2 electrons in its inner shell, and then we have 1, 2, 3, 4, and then if we cover up this oxygen molecule, 5, 6, 7, 8. So there's 8 electrons in this oxygen's outer shell. So it's stable because it has 8 electrons in its outer shell. That's very stable. And if we cover up this hydrogen or this oxygen molecule, we have 1, 2, 3, 4, 5, 6, 7, 8 electrons in this oxygen's outer shell, so it's very stable. Now, when we combine the water to it, we're going to add the hydrogen to those oxygen molecules. Remember, covalent bonding is when we share electrons. If we look at this hydrogen, it's sharing its electrons with oxygen, so it has 2. If we look at, remember, 2 in its outer shell, it's stable. If we look at that hydrogen molecule, it has 2 electrons in its outer shell. It's very stable. And if we look at the oxygen molecule, it has 2 electrons in its outer shell, and then it has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its outer shell. So that water molecule, H2O, that's electrically stable. So we're good there. And the same thing is going to apply to this water molecule because it's the exact same thing. So these molecules are sharing their electrons, the oxygen with the hydrogens. So that is a covalent bond. They're sharing their electrons. All right. And obviously, we don't write all of these. We don't write, these dots and circles and pictures and everything. We actually use structural formulas to show the chemical formula. If we have a single line, that means that there's one pair of electrons that are being shared between those two hydrogen atoms. We go back up here, there's one pair of electrons that are being shared between those two hydrogen atoms. If we have a double or an equal sign, if we have two lines, that's going to represent a double bond. And that means that there's two pairs of electrons that are being shared. So in this case, we have two pairs of electrons that are being shared between oxygen. And we're going to represent that with an equal sign or a double bond. If we look at water, we have a single bond and a single bond to represent the sharing of one pair of electrons. So here we're sharing one pair of electrons between the hydrogen and the oxygen. So we have a single bond. And if we look at carbon dioxide, we'll see that carbon, remember carbon is number 6. So we have 2 in its inner shell, 4 in its outer shell, 4 electrons in its outer shell. So carbon can share 4 electrons. So it would share 1, 2, 3, 4. And then oxygen would share 1, 2, 3, 4. All right, so we're going to briefly talk about electronegativity. Basically, electronegativity is the ability of an atom to attract electrons within a chemical bond. And you can see that electronegativity will increase as we go this way on the periodic table in general. So this element down here will have a low electronegativity. This element up here will have high electronegativity. Basically what that will let us know is that some atoms will have this partial positive charge and others will have this partial negative charge. And it says it will help us determine if this overall compound or overall molecule is polar or nonpolar. All right, so what does that mean? All right, so we have covalent bonding. Remember, covalent bonding is when atoms share electrons. Nonpolar covalent bonds are when electrons are shared equally between those two atoms. Remember those diatomic atoms, N2, H2, O2. They have the same electronegativity. And remember, electronegativity is when the bonds or that those atoms can pull those electrons closer to the nucleus. And we'll see some examples later on. So that would be, those are examples of non-polar covalent bonds. Same electronegativity. Now, polar covalent bonds are found between electrons that are found in bonds that don't have the same electronegativity. So water, we'll look at water. Glucose would be an example. methane, um, so what happens with water, if we go back to our electronegativity, H2O, so oxygen's electronegativity is higher than hydrogen's electronegativity. Oxygen is more electronegative than hydrogen. So we would say that oxygen has a partial negative charge and these hydrogens have a partial positive charge. And when oxygen and hydrogen share electrons, these electrons are actually pulled closer towards the oxygen molecule than they are residing near the hydrogen atom. So we can say that oxygen has a slightly negative charge and the hydrogens have a slightly positive charge. Even though the overall molecule is neutral, because we don't have a H plus, we don't have an O minus or an O2 minus. It's just that due to electronegativity, those electrons are closer to that oxygen atom because of electronegativity. So we would say that oxygen is, or that this bond is a polar covalent bond. It's a covalent bond because the atoms are being shared, or the electrons are being shared between those atoms, but it's a polar covalent bond because they're being shared, but oxygen's kind of hogging the electrons because of oxygen has a higher electronegativity than hydrogen. And that's just really all electronegativity is. And that will kind of help when we talk about polarity. We'll look at polarity when we talk about what can cross over into the cell membrane, what can't cross over into the cell membrane. So when we say polar molecules, that's what I'm talking about. Or polarity, that's what I'm talking about. All right, so remember I said that when we put salt into water, and I should probably move this slide up to right by that question that we talked about earlier. Salt, when we put salt into water, salt is going to dissociate or dissolve into the chloride and sodium molecules. So actually, let me go ahead and move that slide. All right. So I move that slide. Okay. All right. So chemical reactions. Oh, there we go. All right. So hydrogen bonding. This is in one of the questions earlier. And I said that we talk about hydrogen bonding later on. Hydrogen bonding is, I call it a fake bond. It's not really a true bond, but it's almost like that magnet game that I was talking about earlier, that the sodium kind of lines up with the chloride and the positives line up with the negative. But even though we have like this water molecule and this water molecule, what we find is that the water molecules, due to polarity of those water molecules, that we see these hydrogen bonds being formed. And it's usually going to be between that hydrogen end or that slightly positive end. Remember that oxygen has a delta minus and the hydrogens have a delta positive charge, a slightly positive charge, and that oxygen has a slightly negative charge. So that slightly positive charge of the hydrogen compared with a slightly negative end of either an oxygen or a nitrogen molecule of some nearby molecule. Um, um, and just like with magnets, they're going to line up with that positive and negative charge. So the oxygen has that slightly negative, the hydrogen has a slightly positive. So we're going to see this hydrogen bond being formed. It's not a real bond, it's just how molecules kind of orient themselves. We have a slightly negative oxygen here, we have a slightly positive hydrogen here. The molecule is going to orient themselves and create a hydrogen bond. Again, not a real bond. It's just used for an arrangement of molecules. They're going to be formed very easily, broken very easily. If I take a snapshot and then I take another snapshot in a second and in another second, this water molecule might be up against that water molecule, and then another second, this water molecule might be up against that water molecule. So they're going to be formed and broken really easily. These water molecules are going to be moving about. But it's actually these hydrogen bonds are very important in protein structure, nucleic acid structure. This is what gives DNA that double helical structure. We'll talk about the structure of DNA, but I think you all know by now that DNA is kind of coiled and it kind of looks like that ladder shape. Hydrogen bonding is what gives DNA its ladder shape, that coiled double helix shape. Proteins have to have a specific structure in order for it to work. That's what hydrogen bonding is good for. All right, chemical reactions obviously occur when we break chemical bonds or we form chemical bonds or we break those ions up or molecules up. Usually, let's say that we have A plus B we have these reactants. Those are our starting materials. So A and B would be reactants. They can be atoms, ions, molecules. Remember, we saw ions, the sodium ion plus the chloride ion. We saw the hydrogen atom with the oxygen atom yields water. It could also be molecules. And then our products are our finishing thing, whatever we're trying to form. And then this arrow, we say yields. So A plus B yields AB. And then we also have these things called catalysts. Catalysts are basically anything that speeds up the rate of reaction without undergoing any permanent chemical change itself. So if you're in chemistry lab, a lot of times you'll put something over heat to speed up the chemical reaction. Heat is a catalyst. Inside of our body, we rely on catalysts. Typically, if we are talking about something inside the body, we call those enzymes. An enzyme is just a catalyst that's inside of our body. But the key thing about catalysts or enzymes is that that catalyst or enzyme doesn't get consumed. It doesn't undergo any conformational change. So we only need a tiny, tiny, tiny bit of enzyme or catalyst in order to keep that reaction running. And for those of you who have to take organic chemistry, you're going to learn a lot about catalyst. All right, so catalysts. A good example of a catalyst is carbonic anhydrase. So I guess first what I'll talk about is enzymes, or we have this thing called activation energy. So without a catalyst, it's going to take a lot of energy in order for this reaction to happen. But if we throw in a catalyst, it will actually decrease this activation energy. It'll decrease this activation energy so that reaction will, one, take less energy in order to run, and two, make that reaction run faster. So carbonic anhydrase is actually an enzyme found in the human body. It's actually one of the fastest enzymes in our human body. It actually speeds up the reaction between 10,000 and 1 million times faster than it would without the enzyme. But basically, it helps convert carbon dioxide and water to form this thing called carbonic acid. So you can see here carbon dioxide plus water yields bicarbonate plus hydrogen ion going to carbonic acid. But what it does is it helps control pH regulation and fluid balance, and it helps aid in the transport of carbon dioxide. So you'll talk about carbon dioxide a lot next semester in AMP2, but carbon dioxide can be transported in the bloodstream, or when we talk about glycolysis and everything, cells produce carbon dioxide. And somehow we have to get carbon dioxide from the cells back to our lungs to breathe out carbon dioxide. Carbon dioxide can be dissolved in our bloodstream. About 7% of it is dissolved in our bloodstream. We have hemoglobin. Carbon dioxide can be attached to hemoglobin. If that's the case, we call it carbaminohemoglobin. About 23% of carbon dioxide is attached to hemoglobin to get back to our lungs to breathe out carbon dioxide. But about 70% of carbon dioxide is transported through as this method, as this bicarbonate ion. So the majority of carbon dioxide gets back to our lungs through this method of through this enzyme of carbonic anhydrase. Without carbonic anhydrase, your carbon dioxide levels would skyrocket after about 3 breaths, and life would pretty much be unsustainable after about 1 minute of life. So this is just an example of how enzymes work in your body. We'll get into this next semester. All right, so amylase. Amylase is an enzyme that promotes the breakdown of starches during digestion. Which of the following describes the method by which amylase functions? So remember, amylase is an enzyme, so we need to know the definition of an enzyme. So A, it functions as a hormone that signals for starch breakdown to begin. Okay. Maybe we won't cross that off yet because we don't know if the enzyme is a hormone. B, and it inhibits chemical reactions by being changed or depleted by starch. Well, the definition of an enzyme means that it cannot be changed or depleted. So B is wrong. It catalyzes starch breakdown without being changed or depleted. So far, that's my best answer. It gives the definition of an enzyme without being changed or depleted. And it says it catalyzes starch breakdown of starches. It changes its composition in order to break down starch. It cannot change. So my most viable answer is going to be C. And actually, it's not a hormone. All right, you're going to need to know everything on this slide. We have several different types of reactions. And we'll get into more types of reactions later on. But a synthesis reaction. Synthesis just means to add together. Basically, we're taking 1 + 1 = 2. We're taking two smaller parts and we're making something bigger. Growing body parts. A + B yields AB. We're taking sodium and chloride and we're making something bigger. Decomposition, something is decomposing. We're breaking up chemical bonds in order to make something simpler. Breaking up, digestion, absorption of nutrients. We're taking AB and we're breaking it up into simpler components. We're taking hydrogen peroxide and we're making water and oxygen. We're breaking something down. Exchange reactions. Basically, we're breaking something up and we're just switching where things go. Here, we're just replacing where B and D go. Now A and D and C and B are together. Here, we're taking our NO3 and our Cl, and we're just kind of flip-flopping where they go. A reversible reaction. You can identify a reversible reaction with that double-headed arrow. We saw that here with these carbonic anhydrase examples. We have those double-headed arrows. And I'll tell you, most of the reactions in our body happen because of a reversible reaction. It really all depends on what our body needs at the time. In this example, if our body needs more bicarbonate ion, we're going to go this way. If our body needs more carbonic acid, we're going to go this way. It really all depends on what our body needs at the time as to which direction the arrow is going to go. Are we going to go to the right or are we going to go to the left? All right, so the breakdown of table sugar, sucrose, into glucose and fructose is an example of a blank reaction. So your key word here is breakdown. So synthesis. Synthesis means to add together. That's not breakdown. Neutralization, we didn't talk about it. Exchange, that's just the rearranging of something. And decomposition. Decomposition means to break down. So that's going to be our answer. All right, some more definitions. Electrolytes. Those are substances that release ions in water. So we were talking about sodium and chloride that dissociate in water. And I said that dissociate kind of means separate. Also, dissociate means dissolvable in water that I said, but also separate. So sodium and chloride dissociates into sodium ions or sodium cations and chloride anions. But also electrolytes can conduct an electrical current. So that's why we call them electrolytes. So pretty much, why you drink Gatorade is to replace your electrolytes. And if you ever did that experiment in 6th grade where you put the probes in the potato and turn on the light bulb, it's because of electrolytes. Acids. Acids basically are electrolytes that release or dissociate to release hydrogen ions in water. So hydrochloric acid releases a hydrogen ion. Bases are substances that when they dissociate, they can combine with hydrogen ions. And chances are, most of the time, you'll be able to recognize bases because of this OH group. And that OH group would then combine with an acid, such as that H plus, that hydrogen ion. Salts, those are going to be electrolytes formed by the reaction of an electrolyte and a base. So here we have a hydrobromic acid with sodium hydroxide with a base, and we're going to form water and a salt. So in this case, sodium bromide would be the salt. Now, we'll talk about pH later on, but the more acidic an acid is, or the stronger the acid is, the more likely it is it's going to react with a base to form a salt. So battery acid has about a pH of 1, is going to be more likely to react with a base to form a salt when compared to coffee, which has a pH of about 5.0. All right, so acids, substances that release hydrogen ions. So here, typically, you'll be able to identify those because of the H in the beginning of that chemical equation. Acetic acid is different. There's different ways to write acetic acid, but in this case, the H is not first. Bases are any substance that release ions that can combine with that hydrogen ion. And like I said, most cases you'll be able to recognize a base because of that OH at the end. But again, there's an exception. Sodium bicarbonate doesn't have that OH group. And we saw that example up here with this HCO3. That's actually a base because it can combine with that hydrogen ion. And then the salt, remember, is that acid plus a base forms water and a salt. So sodium chloride, obviously, table salt, aluminum chloride, magnesium sulfate, those are all salts. So acid plus a base yields something and a salt. All right, so when we look at acids and bases, we're actually looking at the hydrogen ion concentration of a solution. We can actually stick a probe in the solution, measure how much hydrogen ion concentration there is. When we dissociate that substance, when we put that substance in water, that's that ionic substance that will dissociate. It's going to dissolve. It's going to separate out, and we'll see that polarity due to the electronegativity. So those ions are going to now be surrounded by the water molecules. We'll see those. The positives and the negatives, and the positives and negatives kind of all line up. And now that substance is called an electrolyte. Remember that electrolyte definition can carry an electrical current. So when we dissolve HNO3 nitric acid, it's going to dissociate into that hydrogen ion and that NO3 nitrate anion. So that HNO3 nitric acid, it's an acid. So when like I said, we can measure that concentration of acid based on their hydrogen ion concentration. And not that you need to know, but it's measured in grams per liter body fluid. We use this thing called a pH scale. I'm sure you've heard of. I said pH battery acid of 1.0, pH of coffee is about 5.0. That's just for the shorthand. We actually rarely tell you what the hydrogen ion concentration is. Um, I'll just kind of flip back and forth between this slide and the next slide. So here's your hydrogen ion concentration over here. Rarely will you say, oh, the hydrogen ion concentration is 0.00000001. No one really cares about that. We're gonna translate that into what is the pH. The pH is 8. The pH is 11. The pH is 4. That's easier to understand than, oh, the pH is 0.00000001 or whatever, or the hydrogen ion concentration. So if we look at, the hydrogen ion concentration of 0.01, then the pH is 2.0. So 0.01, the pH is 2. If the hydrogen ion concentration is this, the pH is going to be 9. Basically, we're looking at the number of decimal places after we're looking at the number of decimal places, and that will determine what the pH is. So the lower the whole number on the pH scale, the greater the hydrogen ion concentration. So as we decrease here, we're going to have more hydrogen ion concentration. So here, if we have a pH of 14, we don't have much hydrogen ions. Way down here, we're going to have more hydrogen ions. So it also says the difference in hydrogen ion concentration between each whole number represents a 10-fold difference. So basically, if you go back to math, anytime you move the decimal point over 1, you are either dividing or multiplying by 10. So if we start here and we multiply by 10, we move that decimal point over, multiply by 10, 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 multiply by 10. So this statement right here, that when we go from, every time we go from a whole number on the pH side, that represents a 10-fold difference in the hydrogen ion concentration. All right. Your pH is going to range from 0 to 14. pH of 7 is neutral. Anything that is less than 7 is going to be acidic. It's going to be classified as an acid. Anything above 7 is going to be classified as a base. We're going to say it's basic, or you could also say it's alkaline or alkaline, like an alkaline battery. Now, because I know you want to know, how do we get from this number to this number? So, on your calculator, you have the LN button, the E button, the log button. So if you take negative log of this number, you're going to get this number. You don't have to know that. So negative log of this number will get that number. That's how we measure pH. All right, so pH scale, remember, measures the concentration of hydrogen ions in a solution. Neutral, pH of 7. Basically, it also measures that we have an equal concentration of hydrogen ions and hydroxide ions, OH minuses. This is the pH of pure water or distilled water. Distilled water is what you're supposed to put in an iron, if you've ever used an iron. We've already said that acids have a pH of less than 7. So our hydrogen ions outweigh our hydroxide ions. Vinegar, grapes, tomatoes, coffee, battery acid, beer, butter, white bread. Alkaline, basic, pH of greater than 7. Our hydroxides outweigh our hydrogen ions. Baking soda, bleach, egg whites, milk of magnesia. Here you can see different types of water, tap water, distilled water. I said that distilled water, pure water, is 7.0. Don't memorize this 5.8 to 7.0. Just remember the 7.0. Bottled water can actually have a pH of 5 to 10. Ocean water, about 8. Sparkling water, iceberg water, mineral water. Some people drink mineral water. Some people drink spring water, alkaline water. Just know that there's different types of waters out there. They all have different types of pHs. You don't need to know this slide. So here is different types of acids. I said battery acid was about 1.0. Your stomach acid, your gastric juice, is about 2.0. Distilled water, 7.0. This is the only one that you're going to have to memorize. Distilled water is 7.0. That's neutral. Our blood is about 7.4. We'll learn 7.35 to 7.45. Milk of magnesia, ammonia, household ammonia, sodium bicarbonate, 8.4. So you can just see where different things lie. Caffeine or coffee was about 5.0. All right, so looking at this list, which list only includes acids? Beer, butter, baking soda. So beer has 4.2, so yes, beer is an acid. Butter, 6.1, 6.4, that's an acid. Baking soda, 8.3, no, that's a base. Tomatoes, 4.0 to 4.5, yes. Egg whites, 7.6, no, that's a base. Vinegar, 2.2, yes. Grapes, 3.5, yes. Tomatoes, yes. Coffee, 5.0, yes. That's going to be our answer. Egg whites, that's a base, no. So you're not going to have to memorize any of these things except for 7.0 is pure water or distilled water. Any quiz questions or test questions, you're going to be given a list with the values, and you're going to have to kind of what we did in this question. All right, so our blood ranges from 7.35 to 7.45 with an average of being 7.4. So our blood normally is alkaline or on the basic side. Now we have certain conditions in which our blood has altered pH. We can say that our blood is acidic, or our blood is alkalemic, or that we have a condition called acidosis or a condition called alkalosis. Now, Acidemia or acidosis, or the patient is acidotic, is when the pH of the blood drops below its normal value of 7.35 to 7.45. So here it says acidemia, acidosis occurs when the pH drops below its normal value. Even though it's still on the basic side, it is below our normal value. So the patient is acidotic or is in acidosis, acidemia. Now it says caused by diarrhea, diabetes, lung disease, impaired CO2 exhalation. So we'll get to diarrhea here in a second. Diabetes. Diabetic ketoacidosis. If you ever heard of that term. So acidosis is going to be your key. Lung disease with impaired CO2 exhalation. So we'll learn in A&P2 that your CO2 follows your pH. We kind of alluded to that with our carbonic anhydrase triangle that we saw a little bit earlier, in that anytime your CO2 rises, your hydrogen ion concentration will rise as well. And remember, when your hydrogen ion concentration rises, your pH will fall. If we go back to this side, increase in hydrogen ion concentration, decrease in pH. So with impaired CO2 exhalation, that means we're holding on to carbon dioxide. If we're holding on to carbon dioxide, our carbon dioxide rises, our hydrogen ion concentration rises, so our pH decreases. We become more acidotic. Like I said, I'll come back to diarrhea here in a second. Alkalemia, alkalosis, occurs when our pH becomes too high. We're above 7.45. High altitude breathing, vomiting, high fever, taking excessive antacids. We're not talking like 1 or 2 Tums. We're talking like 9 Tums in the morning, 9 Tums in the afternoon, 9 Tums in the evening. Basically, we're neutralizing our gastric acid, and we're making our body more alkalotic. So excessive antacids. So diarrhea versus vomiting. There's a saying that you lose acid above the belt, you lose base below the belt. So if you're vomiting, you're losing acid. So think about it. If you're vomiting, you're puking up stomach acid. You're losing acid. When you're losing acid, you become more alkalotic. You lose acid, you're vomiting, you lose acid, you become more alkalotic. If you if you have diarrhea, that's below the belt, so you lose base below the belt. So if you lose base, you're becoming more acidotic. So whatever the mechanism, if you're too alkalotic or if you're too acidotic, we do have homeostatic mechanisms to regulate that pH. Everything, we need to be back in homeostasis. We have a lot of different ways that our body protects itself to resist those pH changes. Some of those we'll get into, most we'll get into next semester. Buffers, for example, bicarbonate ions, carbonic acid. Basically, these things are going to either bind to hydrogen ions to help regulate pH, or your kidneys, if you have too much acid, you might pee out some hydrogen ions. Or if you have too much carbon dioxide built up, you might increase your respiratory rate to breathe out more carbon dioxide. All right, so kind of moving on. Organic molecules. Organic molecules always, always, always contain both carbon and hydrogen. Always. If they don't contain carbon and hydrogen, then they are not an organic molecule. And usually organic molecules dissolve in either water or other organic liquids. Now, water-soluble organic compounds, they don't release ions. Remember, if we go back to our periodic table, let's just go back to our periodic table way up here. Organic compounds. Carbon, right here. Hydrogen, right here. What are our electrolytes? Sodium, calcium, magnesium, lithium, potassium. Over here, chloride, bromine, iodine, fluorine. So water-soluble compounds do not release ions. So carbon and hydrogen are not electrolytes. All right, so some examples. Albumin, sucrose, DNA, they're all water soluble. Cholesterol, it's a fat. It's not soluble in water. We have 4 types of organic molecules that we'll talk about. Carbohydrates, lipids, lipids are just fat, proteins, nucleic acids, DNA, RNA. Now, when I say, I'll use the term soluble and insoluble. And I'll use the terms solute and solvent. If you're mixing something in a glass of water, let's say we use the, table salt, you dump some table salt into a glass of water. The solute goes into the solvent. So the table salt is the solute, the water is the solvent. You stir it up, and then your solution is salt water. So the combined result is the solution. So your solute goes into the solvent. The solute is what is being dissolved. The solute or the solvent does the dissolving. So water usually does the dissolving. Inorganic molecules or inorganic compounds do not contain both carbon and hydrogen. So carbon dioxide, water, hydrochloric acid, your ions, electrolytes, sodium, potassium, bromide, iodide, fluorine, those are going to be compounds that ionize in water. Remember, they dissolve in water, they dissociate, they separate. They're going to form ions. Sodium chloride, potassium iodide, all of these dissolve in water. And remember, electrolytes can generate an electrical charge. All right, we talked last chapter about water being the most important substance in the body. About 2/3 of our weight is composed of water, and water is a major component of our body fluids. Remember, water is the solvent. It does play an important role in transporting chemicals throughout the body. Water is a major component of our plasma, of our cardiovascular system. It's going to absorb, transport heat. And then just like with anything in our body, we can, if we drink 3 gallons of water, but we pee out 1 gallon of water, we have a water balance of 2 gallons. Our gains don't equal our losses. A water balance exists, do not. There we go. I should say do not equal losses. Water is a solvent that many solutes dissolve in. I said that last time. Or on the last slide, oxygen. Remember, oxygen used by organelles, specifically the mitochondria, which we'll talk about later on, I think next chapter, to release energy from nutrients, very necessary for survival. All right, carbon dioxide, another inorganic compound, does not contain both carbon and hydrogen. It's a waste product of metabolic reactions of cellular respiration, glycolysis, citric acid cycle, electron transport chain. We'll cover that in chapter 4. It's usually transported as carbonic acid. Remember, I said that there were 3 ways that carbon dioxide can be transported in the body. Dissolved in plasma, attached to hemoglobin as carbaminohemoglobin, and transported as carbonic acid. And then once we get to the lungs, we just exhale carbon dioxide. Inorganic salts. We find a lot of inorganic salts. Those are our electrolytes: sodium, chloride, potassium, calcium. Those are going to play important roles in metabolism. Electrolyte balance exists. No. The balance exists when gains equal losses. So I need to go back up here. The balance exists when gains equal losses. There we go. Okay, now I'm back on the right track. An imbalance exists when gains do not equal losses. Don't know what I was thinking. All right. Balance exists when gains equal losses. An imbalance exists when gains do not equal losses. So we could have an imbalance of water. We could have an imbalance. Let's say you go out for a run and you sweat out. You run 3 miles in the hot sun. And you don't replace any of your fluids or electrolytes. Now you're going to have an imbalance of water and electrolytes. All right, so here's just a table of your ions and inorganic molecules and kind of what they do to help your body. All right, organic substances. Remember we have 4 types of organic substances: carbohydrates, lipids, which are fats, proteins, and nucleic acids. The first type of organic substances that we're going to talk about is carbohydrates. Carbohydrates are the main source of cellular energy, cellular respiration, glycolysis, citric acid cycle, electron transport chain. When we look at carbohydrates, we can dissolve carbohydrates in water. So think of carbohydrates: pasta, bread. If you stick a piece of bread in water, eventually it'll disintegrate. Carbohydrates contain carbon, hydrogen, and oxygen. So it's an organic molecule. It contains carbon and hydrogen. And if we look at the structure of a carbohydrate, we're going to see that the ratio of carbons and hydrogen, or the ratio of hydrogens and oxygens, are very close to 2 to 1. So if we look at glucose, C6H12O6, the ratio of carbons to of hydrogens to oxygens is 2 to 1. 12 to 6 is 2 to 1. When we break down carbohydrates, or we actually have 3 types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides, we classify those as simple sugars. Basically, they are one sugar molecule. They all have the same similar structure of C6H12O6. Um, and saccharide is just another term for sugar. But we have, depending on how they're arranged, how the carbons, hydrogens, and oxygens are arranged, we have three different types of monosaccharides: glucose, fructose, and galactose. If we have a disaccharide is just two sugar molecules attached to each other. We have three different types of disaccharides: sucrose, lactose, and maltose. A polysaccharide, we say that those are complex carbohydrates. Basically, those are going to be three or more sugar molecules attached to each other. Now, rarely are we going to see three sugar molecules. We're going to see more up to hundreds or thousands of sugar molecules attached to each other to create a polysaccharide. Starch, glycogen, cellulose. Glycogen, we'll talk a lot about glycogen later on, but I'll always say glycogen. Glycogen's a polysaccharide because it has many sugar molecules attached to each other. But glycogen is actually stored glucose that's found in your liver and your skeletal muscle cells. So for those of you who do carb loading the night before a big swim meet or a track meet or whatever, that you need energy the next day, the excess carbs would get stored as glycogen in your liver and skeletal muscle cells to be used for energy the next day. And that's just what glycogen is. All right, so this is just a picture example of a monosaccharide. It's just one sugar molecule. Disaccharide, two sugar molecules. Polysaccharide, bunch of sugar molecules. All right, lipids. Lipids are fats. They're insoluble in water. They do not dissolve in water. Remember, oil and water, they don't mix. So lipids do not dissolve in water, but they are soluble in other organic solvents. We'll talk later on about how lipids are transported in the bloodstream, because your bloodstream contains water. And since lipids can't be around water, lipids have to be packaged. We call that emulsification. So they have to be packaged in this special way in order for them to be transported in the bloodstream. So we have different types of lipids. Triglycerides are going to be your most common. We have these things called phospholipids, steroids. We'll see a lot of these in our cell membranes. But we also have, they also play another other functions in cells. So triglycerides, again, most important lipid. We use, like I said, we use carbs primarily for cellular energy, but we can also use fats. Typically, we'll use fats for energy when we do light activity or when we're sleeping. Now, if you ever take nutrition, you'll learn that carbs and proteins have 4 calories per gram and fats have 9 calories per gram. So if you're eating, let's say, if you're eating, 1 ounce of, let's say you're eating 3 ounces of protein versus 3 ounces of fat, you're actually getting over twice the amount of calories from eating the 3 ounces of fat than you are from eating the 3 ounces of protein or the 3 ounces of carbohydrates, which you'll learn about nutrition. So fats or triglycerides contain carbon, hydrogen, and oxygen, just like carbohydrates do, but they don't have that ratio of 2 to 1 hydrogens and oxygens like carbohydrates do. And we abbreviate carbohydrates as CHO. I don't think I said that before. So carbohydrates are abbreviated CHOs. When we look at the structure of fats or triglycerides, we'll see that we have one glycerol molecule and 3 fatty acids or 3 fatty acid side chains. And this triglyceride, it's important later on, but this is it does not have a polar region. So we'll talk about that later on when things cross over the cell membrane. So no polar region in triglycerides. All right, so we have different types of fats. You've heard of saturated fats, unsaturated fats, polyunsaturated fats, trans fats. Basically, earlier on we talked about single bonds and double bonds. So saturated fats only have. So this is our. We go back up to our structure here. We have our glycerol portion, and then we start out with our fatty acid side chain with that carbon-oxygen double bond, and then we have our fatty acid side chain. So that's kind of where we are right here. And then from here on out, in a saturated fatty acid, all we see are single bonds from these carbons, these carbon-carbon single bonds. So I think of saturated fatty acids as saturated with single bonds. So they're saturated, they have all single bonds between these carbons. So they only have carbon-carbon single bonds. Most of your saturated fatty acids are going to come from animal products, going to be solid at room temperature. Regular butter is solid at room temperature. Typically, these are going to be worse off for you from a cardiovascular standpoint than unsaturated fatty acids. Now, unsaturated fatty acids have at least one or more carbon-carbon double bond. And we can see a double bond here. We can see a double bond here. So we have at least one double bond. If we had only one double bond, we would say that it's a mono unsaturated fatty acid. If we have 2 or more double bonds, we would say it's a poly unsaturated fatty acid. Typically, your unsaturated fatty acids are going to be of plant origin, liquid at room temperature, better for you from a cardiovascular standpoint than saturated fatty acids. Your olive oils, your avocado oil, your saffron, your saffron oil, sunflower oil, things better for you cardiovascularly. Now, without getting into too much organic chemistry, you see how we have our carbon-carbon double bond, and then we have the H up here, and then we have the H up here. So we're looking kind of right here. When we get into organic chemistry, that would be what's called a cis conformation. If we had our carbon-carbon double bond and we had one H up here and one H down here, that would be a trans conformation. So even though So this would be like your trans fat. Trans fats are, even though they're considered an unsaturated fatty acid, these are going to be the worst for you cardiovascular wise. These are your things that taste really good. Your fried chickens, your fried foods, things that are fried in trans fat. So this is the one exception to the rule that unsaturated fatty acids are good for you from a cardiovascular standpoint. So you don't have to know the organic chemistry, between cis and trans and where the hydrogens lie, but just wanted to point that out to you. All right, so lard, it's a fat that is solid at room temperature, will have blank than peanut oil. So will it have more oxygen atoms? We go back to our structure. Well, we don't see any oxygen atoms in our fatty acid side chain, so we'll cross that one off. It will have fewer hydrogen atoms bonded to carbon atoms. It will have more glycerol groups. Well, we go back up here. We only have one glycerol group, so we can throw out that. More carbon-carbon single bonds. So we go back to our saturated versus unsaturated. Saturated says, wrong way. Lard is a fat that is solid at room temperature. Solid at room temperature will only have single bonds. Peanut oil will have double bonds. So something that is solid at room temperature will have more single carbon-carbon bonds than peanut oil. So that's going to be our answer. And I really didn't address B. Only because I wanted to get to D as the right answer. If we look at, if we actually count the number of hydrogens up here versus the number of hydrogens down here, we actually don't know how many hydrogen atoms lard will have versus peanut oil, because we don't know the structure of lard and we don't know the structure of peanut oil. So we don't know how many hydrogen atoms there are going to be. But we do know that lard is going to have more single carbon-carbon bonds, or lard is only going to have single bonds compared to peanut oil, which is a liquid at room temperature, which is going to have double bonds. All right, lipids, continuing on, we talked about triglycerides. Now we're going to talk about phospholipids. Phospholipids still have that glycerol backbone, but they have two fatty acid side chains instead of three. And in place of that third one, they have that phosphate group. We see a lot of phospholipids in our cell membrane. We have about 1,000 different types of phospholipids out there. Another type of fat or another type of lipid is steroids. This is very structurally different from the rest of the fats that we've seen. They have 4 rings to them. So 4 connected carbon rings, again, component of cell membranes. They synthesize a lot of our adrenal and sex hormones, testosterone, estrogen, adrenal hormones, epinephrine, norepinephrine. So cholesterol is going to be the main steroid in the body. Cholesterol is lipid soluble. Remember, cholesterol is a fat. Lipids are fats. That's what we're talking about. So cholesterol is not soluble in water. Remember that phospholipid, if you remember that phospholipid bilayer of the cell membrane, that's what we're talking about here, that phospholipid bilayer. So we have our triglyceride with our glycerol portion and our 3 fatty acid side chains. Our phospholipid has that glycerol portion, 2 fatty acid side chains, and that phosphate group. That's what's going to make up our phospholipid bilayer of our cell membrane. So remember our fatty acid, and we'll talk about this later on when we talk about the cell membrane. Fatty acid, we have 2 fatty acid side chains. Remember, fatty acids are not soluble in water. So they are water insoluble. So that's why we have a water insoluble portion of our cell membrane. And then that glycerol portion is going to be soluble. So we have a soluble portion of our, or that phosphate portion is going to be soluble in water. All right, so general structure of a steroid, remember, has 4 rings. And this is the structure of cholesterol. You're not going to have to know that, but I just wanted to show you that. All right, proteins. We have over 2,000 different types of proteins. Antibody proteins. We have hemoglobin, myoglobin. They both carry oxygen. Hemoglobin carries oxygen throughout the bloodstream. Myoglobin carries oxygen in the muscles. We have structural proteins, or their proteins are used as structural materials inside the cell. We use proteins as an energy source. We use proteins as chemical messengers and hormones and receptors, enzymes like catalysts. Receptors allow cells to respond to specific types of molecules that bind to them. Like blood typing, or we have, glucose receptors, so that glucose can bind to those receptors and bring glucose into the cell, or sometimes a catalyst or an enzyme will bind to that receptor so that some specific function can happen to that cell. I'm sure you've all heard amino acids are the building blocks of proteins. If we look at the structure of an amino acid, we're going to kind of see 3 different components. We're going to have, or 4 different components. We're going to have this central carbon right here. We're going to have this amino group, this NH2 group. We're going to have this carboxyl group, this COOH, and then we're going to have this this R group, and I'll go down here to this R group. And in this case, R is going to equal H. Depending on what amino acid we're talking about, this R group would be one of many different things. We have 20 amino acids, so this R group would represent one of 20 different things. And in this case, R would equal H. And if we look at here, this R group would equal that. In this case, this would be cysteine. R would equal this. In this case, this is phenylalanine. And this 3-dimensional shape, or that conformation of a protein molecule, is very critical to the function. I talked earlier about hydrogen bonding being very critical to the function of DNA structure and being very critical to protein function. And if the protein is not folded correctly or is not shaped correctly, that protein might be rendered useless. Now, amino acids, when we combine amino acids, basically amino acids are connected by this thing called a peptide bond from this carbon and this nitrogen, the carbon of one amino acid to the nitrogen of another amino acid. All right, so an amino acid is a polar molecule. They have polar regions. So remember, I told you that hydrogen bonding forms typically between oxygen and between the hydrogen and either nitrogen or oxygen. So we're looking at the nitrogens and the oxygens versus that hydrogen. And then there's our peptide bond right there. When we look at the structure of a protein, we can classify the structure of a protein on 4 different levels. Usually, sometimes 3, sometimes 4 different levels. Basically, remember, amino acids are the building blocks of protein. The primary level of protein structure is just the sequence of amino acids. Remember, we have 20 amino acids. And just that sequence of amino acids is that primary structure. And proteins can be anywhere from 100 to 500 or 5,000 or more amino acids long. And remember, I told you that Titan was what, 34,350 amino acids long. Your secondary structure of proteins is formed by hydrogen bonding. Remember the hydrogens versus those oxygens and nitrogens. And what happens is that those hydrogen bonds will actually form these things called alpha helices, an alpha helix, or these beta pleated sheets. So this alpha helix right here is going to kind of coil up almost like that DNA, or these beta pleated sheets, which is basically like you fold a piece of paper over and over and over and over. And that's what a beta pleated sheet is. It's just like folding a piece of paper. Keep folding a piece of paper. So that's your secondary protein structure. Your tertiary structure is how that protein is represented in a three-dimensional view. It's a three-dimensional version of that protein. And that's going to determine that protein function. We have these things called chaperone proteins that play a role in shaping the protein. So if your protein is malfunctioned because of its shape, chances are it's going to be because of a malfunctioning chaperone protein. Now, I said that we either have 3 or 4 levels, the tertiary or the quaternary structure of a protein. If we can have we can either stop at the tertiary level or we can move on to the quaternary level. If we have multiple proteins that come together to form one macro protein, then we can call that a quaternary structure. If we're looking at hemoglobin is actually formed by 4 proteins coming together to form one large macroprotein. So a quaternary structure would be 2 or more polypeptide chains or 2 or more tertiary proteins coming together to connect itself into one large macroprotein. So here, primary structure is just the sequence of amino acids. Your secondary structure, alpha helices, beta pleated sheets, due to hydrogen bonding. Tertiary structure, how is that protein folded in a three-dimensional world? Usually due to those chaperone proteins. And then if we have a quaternary structure, those are going to be multiple proteins coming together, or multiple polypeptide chains coming together to form one large macro protein. All right, so complete ribosomes found within cells of the body. Ribosomes are actually an organelle, which we haven't talked about yet. That's next chapter, but you should have already had that in Bio 1000. So complete ribosomes found within cells of the body are formed by the association of different protein subunits. Each complete ribosome represents the blank of this organelle: primary, secondary, tertiary, or quaternary. So to me, this should be a gimme question. So first thing, complete ribosomes, different protein subunits. Organelles. So if we go back to chapter 1, and we learned about, atoms, molecules, or atoms, elements, molecules, macromolecules, organelles. So organelles are bigger than macromolecules. So they're really big. But different protein subunits. So basically, we have a protein here. And if we're taking different protein subunits and putting them together to make an organelle, we have to be talking about a quaternary structure. All right, so one disease, it's a genetic disease called cystic fibrosis. Basically, it's a mutation of this thing called CFTR, cystic fibrosis transmembrane conductance regulator gene. It's where the protein, this regulator protein, gets misfolded. And it leads to a buildup of chloride anions inside the cell. And what happens is, the cell wants to maintain kind of neutrality. So because we have chloride inside the cell, it pulls sodium inside the cell as well. And one thing that you'll learn, or that you need to know, is that wherever sodium goes, water will go as well. So water follows sodium. You'll definitely know this when you get to the renal chapter next semester. Inside the lungs, your mucus becomes real thick, sticky. It's very hard to clear that mucus. It's very hard to cough. And these patients often have difficulty breathing. They get infected very easily because that mucus gets trapped in their lungs. And a lot of times these patients have, it's called percussion therapy. Usually as infants, you kind of put them on their stomach and you kind of beat their back a little bit. Or as they get older, they'll wear these vests, these percussion vests, that'll kind of thump their chest to break up that mucus so that they can cough it up. All right, so there are ways that proteins can get denatured. And denatured or denaturation just means to take away or alter the qualities of, the natural qualities of. And typically, when we denature proteins, we're talking about the secondary or tertiary structure of that protein. Um, and typically, these are bad things. Um, Remember that secondary structure is your hydrogen bonding, so your alpha helices, your beta pleated sheets, and that tertiary structure is that overall three-dimensional structure of based on chaperone proteins. But your primary structure of the sequence of amino acids, that's going to remain intact. So the composition or the amino acid sequence of that protein remains intact. And proteins are denatured because of heat, radiation, pH changes, chemicals. Sometimes this is reversible, other times it's not reversible. And the body is sometimes good about repairing the proteins. Sometimes the body is not good about repairing proteins. Sometimes it'll just scrap the protein and say, you know what, it's too much energy to repair this protein. Let's just scrap it and start all over. And the body will kind of recycle those proteins and start over. But a good example of this is not that we see a lot of people who get perms anymore or permanents, but people, when you get your hair curled, or people that use curling irons, you're actually denaturing the proteins when you curl your hair. All right, nucleic acids, deoxyribonucleic acid. DNA. Remember, DNA is double-stranded. Ribonucleic acid, RNA, it's single-stranded. Remember, both undergo replication. We'll talk about mitosis later on in a subsequent chapter, and we'll talk about meiosis at the very last chapter in AMP2. But basically, these nucleic acids are comprised of these building blocks called nucleotides. And these nucleotides have this sugar group. And if we're talking about DNA, this sugar group is called deoxyribose. In RNA, it's called ribose. We also have this phosphate group. And we also have this organic nitrogenous base. So if you remember adenine, thymine, guanine, cytosine, or uracil, those are your 5 bases. This is just kind of like your structure of DNA and RNA, kind of at the 2-dimensional level. Remember and we'll talk about this in a subsequent chapter, but your A's are always going to pair with your T's. Your C's and G's are always going to pair together. And then on the RNA side, and like I said, we'll talk about this later, we don't have any T's or thymines in RNA. We only have U's. The T's are replaced with U's in RNA. All right, so DNA codes for your genetic information. I said it has that sugar deoxyribose. It's double coiled. It has that double helix structure. Its 4 bases are A, T, G, and C. Adenine, thymine, guanine, cytosine. Has that phosphate group as well. RNA takes that DNA code and uses that code to synthesize proteins. That's the function of RNA. It contains the sugar ribose. It's only single-stranded and contains AUGC. adenine, uracil, guanine, cytosine. Remember that thymine is not present in RNA. It's uracil. Also has that phosphate group as well. Ribose, RNA, deoxyribose, DNA. The only difference between deoxyribose and ribose, deoxy, right there. Deoxy is lack of oxygen, right there. All right, so kind of one or two more slides to go, and then we're done. So CTs, computerized tomography imaging. Basically, it uses two-dimensional pictures or two-dimensional x-rays to create a three-dimensional image of soft tissue. It helps visualize internal anatomy, helps looks at different densities, so it can help identify tumors. Positron emission tomography or PET scans. It actually uses a radioactive isotope like carbon-11. It emits these things called positrons. Now, we haven't talked about positrons. We've talked about protons, electrons, and neutrons. Remember, protons are positively charged, neutrons have neutral charge, and electrons have a negative charge. Now, positron is actually a positively charged electron. And we're not going to go into how that happens, but PET scans use positively charged electrons called positrons. Has a characteristic of electron, but it has a positive charge. Oops. There are my dogs. All right. Okay, so an individual with a condition called phenylketonuria cannot break down the amino acid phenylalanine. Molecules that include phenylalanine will build up in the blood, potentially causing intellectual disability and other symptoms. This inherited disease can be controlled by following a diet that is low in what? Nucleic acids, carbs, cholesterol, or protein. So, an individual with the condition called phenylalanine cannot break down the amino acid phenylalanine. You don't need to know anything about phenylketonuria. You don't need to know anything about phenylalanine. All you need to know is amino acid. This inherited disease can be controlled by following a diet that is very low in what? Amino acids are the building blocks of proteins. So if you follow a diet low in protein, then you're not going to get that many amino acids. So that would be your answer. All right, so that is the end of chapter 2, very long chapter. So please let me know if you have any questions. Take care, and I will see you next chapter. Bye.